started their life from single cell. How it is possible, let us see. As we know that sperm cell nucleus combines with ova, gives single cell structure, that is diploid cell called zygote. Okay. How this single cell called zygote undergoes for further developments and further steps, and how it helps in the process of formation of a complex large organism. Okay. And what are the various steps that are there in the growth, and all these things we are going to see in the living organisms. All cells are reproduced by dividing into two. Let us take a single cell. Imagine it is a single cell. It is divided into two daughter cells. Each daughter cell can become a parent cell in the coming generations. So which gives rise to two daughter cells each time they divide. These nearly formed daughter cells, and the daughter cells also grows, undergoes for division, undergoes for development. Okay, which leads to formation of new cell population that is formed by the growth, and division. And here, in other words, such cycles of growth and division allows a single cell to form a structure consisting of millions of cells. Here in this class, let us see the first type of cell division called mitosis. Mitosis involves various stages, such as 4 major stages are there in the mitosis. First one is prophase, second one is metaphase, and the third one is anaphase, and the fourth one is telophase. So at the end of the telophase, we are going to see formation of two daughter cells from a single parent cell. Okay? Mitosis and G1 phase, G2 phase, gap 2 phase. In between these two phases, S phase is also present. Now here, if you carefully observe the cell cycle, cell division is a very important process. During the division of a cell, basically DNA replication occurs, cell growth occurs, or generally takes place. All this process, even including cell division, DNA replication, etc., and cell growth, and hence have to take place in the coordinated manner or way to ensure correct division of the formation of progeny cells. What are they? Progeny cells intact into the complete genomes. Sequence of events by which a cell duplicates. Sequence of events by which cell duplicates its genetic material called genome. Synthesis that occur constitute the cell and eventually divided into two daughter cells. Two daughter cells is termed as a cell cycle. Although, if you carefully observe the cell growth, it is a continuous process. DNA synthesis occurs only during the time of specific stage, okay, in the cell cycle. Here, the replicated chromosomes, whatever there, the replicated DNA that is present in chromosomes are then distributed to various daughter cells, okay, or daughter nuclei. The cells contain nuclei by a complex series of events during cell division. The events that are themselves undergoes to genetic control. Now let us take one example here. A typical eukaryotic cell is there. What is eukaryotic cell? Nothing but cell with a proper visible nucleus. which is surrounded by double membrane structure. Hence it is called eukaryotic cell. It is illustrated by human cells in culture. Cells divide once in approximately 24 hours once. Okay. However, during this duration of cell cycle can vary from organism to organism, also from cell to cell type. Let us take yeast, for example, yeast cells or yeast organisms progress through the cell cycle only about 90 minutes of time. How much time? 90 minutes of time. The cell cycle is divided into two basic phases. Okay. First phase is interphase. What is that? Interphase. And the second phase is called, okay, the interphase and the M phase, mitosis phase, and other phases. Okay. And here, after this phase of cell division, what we have to learn? Here in this diagram also you can see, okay, after M phase you can see G1 phase, G2 phase and S phase. Right? Now, it is significant to note that in the 24 hours duration of time, duration of cell cycle of human cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of cell cycle. Okay. So, in the entire cell cycle, 95% of the cell cycle contain an interphase. Right. M phase starts with nuclear division, corresponding to the separation of daughter chromosomes. Okay. And usually ends with the division of cytoplasm. Now here, corresponding to the segregation of daughter chromosomes, karyokinesis, which is nothing but division of nucleus, and cytoplasm division. If you take the cytoplasm division in the cell during cell division, it is called cytokinesis. Okay. Now here, the interphase, so called resting phase, is the time during interphase is called resting phase, which the cell is preparing for division by undergoing both cell growth. Okay, during interphase cell growth occurs, DNA replication occurs in orderly manner. The interphase is divided into three further phases. What are they? First one is here Z1 phase, that is the first phase, and the next one is S phase, that is the next phase, and the next phase is G2 phase. Okay, gap 1 phase, gap 2 phase, and the S phase, synthesis phase. Okay, now here what happens during G1 phase? Cell metabolically active and continuously grow during G1 phase, but does not replicate the DNA. Or synthesis phase marks the, if you take the S phase, synthesis phase marks the period during which DNA synthesis generally takes place. Apart from this, during this time, amount of DNA per cell generally increases or doubles. Here, if the initial amount of DNA in the beginning of the cell, whatever the amount of DNA is denoted as a 2C, then it increases to 4C. Okay. However, there is no increase in the chromosome number. Chromosome number will not increase. Chromosome number will not increases, but the amount of DNA will be increases. Okay. If the cell had diploid condition, okay, which means J-code, which is 2n condition, number of chromosomes at G1 phase will be, even after S phase will be, number of chromosomes remain same. Okay. 2n in animal cells during the S phase, in animal cells during S phase, DNA replication begins in the nucleus. Okay, and the centriole duplicates in the cytoplasm. And during G2 phase, what will happen? You see here, gap 2 phase, protein synthesis. What is that? Proteins are synthesized in the preparation for mitosis. while the cell growth is continues. While the cell growth continues. Okay. Finally, some cells in the adult animals do not appear during cell division, and many other cells divide only occasionally as they needed to replace. These cells that do not have divide further at the G1 phase, to enter into inactive stage called quiescent stage or G0 phase. G0 phase is an inactive phase where cell division will not occur in the specific cells. Cells in this stage remain metabolically active but no longer proliferates. The process of proliferation will be absent in these cells unless called on to do some depending on the requirement of organisms. In animal cells, mitotic cell division is only seen in the diploid somatic cells. What are diploid somatic cells? What are diploid somatic cells? Let us take the brain cells. They are diploid somatic cells, muscle cells, bone cells. However, there are few exceptions to this. If you take the haploid cells like sperm cell and ova, they are divided by mitosis, for example. Male honeybee, if you take male honeybee, okay, against this, as you know that they divide by mitosis, okay, male honeybees. Now, against this, the plants can show mitotic cell division. Plants can show mitotic divisions in both the haploid and the diploid cells. From your recollection, examples of alteration of generations in the plants. Identify the plant species and stages at which mitosis is seen in the haploid cells. Remaining